Our final speaker of the session is Victoria Richon. Victoria received her PhD at the University of Nebraska and went on to do postdoctoral research at Memorial Sloan Kettering Cancer Center. She has had an illustrious history of working in industry and developing therapeutic targets. Perhaps most well known is that she was a lead member of the scientific group that discovered the histone deacetylase inhibitor, SAHA. And so this discovery was actually the basis of Aten Pharma, a company that Dr. Richon co-founded and where she serves as an executive director. So there's a number of things that she's been involved with development, and she continues this as Vice President of Biological Sciences at Epizyme, where she joined in 2008. And so I think her goal remains the same, to move this field forward, to discover and develop small molecule histone methyltransferase inhibitors for personalized therapeutics. So I look forward to hearing your talk.
Well, thank you, and I want to thank Tyler and Jackie and Rudy for inviting me, and it's really great to be the final speaker before lunch, and always to follow Jay's really inspiring talk. So what I'm going to talk to you today about is our work on the development of methyltransferase inhibitors. And so first I'm going to give a little background on the histone methyltransferases and tell you two stories of our lead program, one in targeting DOT1L and the second in targeting EZH2 in specific cancer subtypes. So first, a little background about histone methyltransferases. And so there are about 96 members of this family in humans that share a similar catalytic domain. They all use a common nucleotide substrate for transferring the methyl group, that's S-adenosylmethionine. The other thing that was important to us as we were beginning our drug discovery efforts was that we knew the substrates for many of these enzymes as well. And we think that there's a limited number of substrates for many of these enzymes, leading to biological specificity. The members of this family can be divided into two subfamilies, the lysine methyltransferases and the arginine methyltransferases. The lysine methyltransferases are mainly the set domain family, and interestingly, this is S-adenosylmethionine, and in all of these, they share a similar catalytic domain, and you can see that on the basis of the structure of SAM in the catalytic domain. Whereas the arginine methyltransferases form a family as well of about 50 members, and they also share a similar catalytic domain, as well as having the same structure of SAM, similar structure of SAM binding within their catalytic domain. Interestingly, there's one exception to the rule as being an arginine methyltransferase, and that's the lysine methyltransferase DOT1L. That's a non-set domain methyltransferase that also shares the same similar catalytic domain and the binding of SAM in its catalytic domain. So we also became interested not just based on the druggability of this family, or potential druggability, but also because many of these members, as Jackie talked about in the beginning introduction, actually show alterations in cancer. And when we started Epizyme 4 years ago, as is today, this was really a rapidly evolving area of biology, and as has been mentioned many times, that with the understanding of all of the genetic alterations in cancer, that this family of enzymes is a family that keeps popping up as having alterations in cancer. And today, as when we started the company, we thought this was wide open because there are currently no compounds in clinical development. So just a little more, oops, I don't know where that is. Okay. Just a few representative genetic alterations in the methyltransferases as well as the demethylases in cancer. And so the genetic alterations can either have been found to be either translocations in the case of MLL, and also translocations in the case of WISC1 or MMSET in the 414 subtype of myeloma. as well as amplifications. Specifically in EZH2, there's also amplifications and mutations. And it's not just in the histone methyltransferases at the top of the slide, but you also see the methyl mark being altered by the demethylases as well, being mutated in cancer. And just two examples of that, where you see UTX being mutated and inactivated, where you also can have gain of function with GASC1, in which it's been shown to be amplified in specific cancers as well. So for the purposes of the talk today, I'd like to focus, as I said, on two examples that we've been working on, and that's focusing on targeting DOT1 in the specific subtype of leukemia that has the translocation of MLL, and then I'll also talk about our efforts in EZH2. So background on DOT1. So DOT1, as I said, is a non-set domain histone methyltransferase. It targets histone H3, lysine 79, and it can mono-di and trimethylate H3K79. It's the only known methyltransferase that catalyzes methylation of H3K79. As Rick talked about, the histone H3K79 methylation is associated with actively transcribed chromatin. And when Yi Zhang actually identified DOT1L as a histone H3K79 methyltransferase, one of the first experiments that he did was a two-hybrid analysis in which he asked what proteins bind to DOT1L. And interesting, what he found was AF10 as the interacting protein. And so that really opened up the biology and studying the biology of DOT1L. And when he looked to see what other associations in addition to transcriptional elongation complexes that these proteins are involved in, he also found disease relevance, and that is one of the translocation partners for this MLL and MLL rearranged leukemia. So there's many studies now have been done, and I think Scott Armstrong is going to talk a little bit more about this later on, showing the genetic role of DOT1L in these MLL rearrangements. I'll just give a little more background here. So MLL is a histone methyltransferase itself. It catalyzes histone H3K4 methylation, and it turns out to be rearranged in about 5% of acute lymphocytic leukemia and about 5 to 10% of acute myelogenous leukemia. A very important subset in that it's rearranged in 70% of infant acute leukemias. Also, interestingly, over 80 fusion partners have been described. And these, because we've been able to identify these using cytogenetics and looking for 11q23 alterations, these patients are known to have a poor prognosis. So what happens when you have the translocation is that you retain in the fusion the DNA targeting domain, but you actually lose the methyltransferase or set domain. And so you form these fusions that have the DNA targeting domain and then these other proteins that are all known to be involved in this DOT1L complex. So what we think is happening through a lot of beautiful work that's done by many, including Scott Armstrong, Jay Hess, as well as Rick Young's lab, is that we know that MLL is recruited to specific areas of the chromatin and can methylate H3K4. When you now have the fusion, you see that you lose the set domain, so you no longer methylate with the fusion H3K4 methylation, but you now recruit in DOT1L, and you see aberrant localization and methylation of H3K79. And this is work from Scott's lab, where he's been able to map through ChIP-seq the aberrant H3K79. What's also known, because this subset of leukemia has been studied, is that there's a very distinctive transcription profile associated with this type of leukemia. And what Scott did was to look at the correlation between the aberrant methylation and saw a very nice overlap between those genes that are upregulated in this type of leukemia, in the MLL rearranged leukemia. And so it's this aberrant gene expression pattern that is then believed to lead to leukemogenesis. And there are two genes that we've been focusing on in our work to specifically look at as probes, HOXA9 and MEIS1. And so this was a really nice project for us because there have been several genetic studies showing a really important role for DOT1L in this MLL rearrangement. But we wanted to now make a small molecule DOT1L inhibitor and ask whether this is going to be a good therapeutic intervention for MLL rearranged leukemia. So we took multiple approaches to do this because there was really no precedent for small molecule selective inhibitors. So we did screening as well as mechanism-guided design. And where we made the most traction was in the mechanism-guided design. And this is a small molecule that we've identified that is a very potent DOT1L inhibitor. It's a SAM mimetic. And so you can see that it's a sub-nanomolar inhibitor of DOT1L enzymatic activity. Very importantly as well, it's a selective inhibitor. These are different, this is an IC50s for different enzymes that are methyltransferases that are both the lysine methyltransferases and the arginine methyltransferases. And you see that there's greater than 1,000-fold selective for DOT1L inhibition. Also, the next question we asked was, is it selective in inhibiting H3K79 methylation in cells? And so you can see a very nice dose-dependent inhibition of H3K79 dimethylation. These are two different MLL rearranged cell lines that either have the MLL-AF4 or MLL-AF9 translocation. And you can see a nice diminution in both of those cell lines after 4 days of treatment. We also asked about selectivity in cells. We saw nice selectivity in the enzyme assays. Do we also see selectivity in cells regarding effects on other methyl marks following treatment with the inhibitor? And you can see that treatment with 10 micromolar, which is a high level of the inhibitor in cells for 4 days, that we only see effects on H3K79 methylation, and we see no effect on any of the other either lysine or arginine methyl marks that we examined. So the next experiment that we did was to say, following the mechanism, we would expect to see a decrease in following the decrease in H3K79 methylation, an effect on these target genes. And so we can see a nice effect and decrease that's time-dependent on HOXA9 and MEIS1 gene expression. We don't see effect on all genes, and this is just shown here by TBP, where we don't see any effect on TBP. And when we've done gene expression profiling experiments, we've been able to see that we really are specifically reversing that gene signature that I showed you on the other cell. And that in gene set enrichment analysis is one of the highest gene sets that we see in terms of changes in expression and correlation. So one of the questions that we're often asked is, but do the cells care? And so we now looked and saw what happened over time with proliferation in MLL rearranged cells and also in non-MLL rearranged leukemia cells. So these are the MV411 and the MOM13 cells that I showed you on the last slide. And you can see, consistent with the mechanism where we first see the decrease in H3K79 methylation, then decrease in the genes, that it takes about 7 days before we start seeing an anti-proliferative effect in both of these cell types. Whereas in a non-MLL rearranged cell line, we don't see that dependency on DOT1L. And so even though we're affecting and decreasing H3K79 methylation, we really see no effect on proliferation of these cells. We've also expanded this panel out to look at multiple cells that have the rearrangement versus other cells leukemia cells that do not have the rearrangement. And we see a nice correlation with sensitivity with those cells that have the rearrangement and all of the different rearrangements, including, I showed you before, AF4, AF9, as well as ENL, which are all expected to recruit in DOT1L as part of the complex. Whereas those cell lines that don't have the rearrangement, we really see much less sensitivity. So these cells are really dependent on DOT1L for their viability and proliferation. So the next experiment that we wanted to do was, like Jay said, really the litmus is, can we treat animals with these inhibitors and see an anti-tumor effect? And so we created a xenograft model, in this case in rats, and so in which we treated those animals for 21 days with the .1L inhibitor, and you can see the vehicle compared to 2 different doses of the inhibitor during that 21 days. You can see a very dramatic anti-proliferative activity, tumor stasis, as well as at the highest dose, tumor regressions. And very interestingly, when you stop dosing, you can see at the higher dose, where we see the regressions, you see no regrowth for up to 50 days. So these tumors are, we see a complete regression. At the lower dose, you see a nice regression, but then when you stop dosing, you see regrowth. Also importantly, there's no effect in body weight at these 2 doses, and we knew that the inhibitor was hitting and inhibiting .1L activity because we can see a nice effect and decrease on H3K79 methylation at the 2 doses that were delivered. So what I've shown you today about .1L is that we've been able to make .1L inhibitors that potently block the enzyme activity and decrease methylation selectively at H3K79, reverse the MLL-driven gene expression signature, and selectively kill the MLL-rearranged cells, both in culture and in xenograft models. And so we're very excited about moving this program forward and testing these hypotheses in patients that have the MLL rearrangement. I'd like to next move on to a second story about EZH2. And so EZH2 has been discussed as the catalytic subunit of the polycomb repressive complex, or PRC2, which has been talked about early and is involved in development and cell fate. EZH2 is a set domain histone methyltransferase, and it targets histone H3 lysine 27, and it can mono, di, and trimethylate this site. I usually refer to it as sort of the rock star of the methyltransferases as its involvement in cancer, in that there are many, many papers on showing that it's overexpressed or amplified in many human tumors, and that's in many different solid tumor types. There are a large number of papers also showing that it has oncogenic activity in cells and in vivo, and importantly for us, as we're thinking about making catalytic inhibitors, that the catalytic activity is required in some of the studies showing that for its oncogenic activity. Also, there have been many studies showing that genetic knockdown can impair tumor cell growth, and that's both in cells and in animals as well. We became very interested in EZH2 when we also saw that there was a paper showing that there were mutations in EZH2 identified in non-Hodgkin lymphomas. And this was work from Mark O'Mara's lab at the British Columbia Cancer Agency that was published early in 2010. And what he found was that the mutations were identified in specific subsets of non-Hodgkin lymphoma, and that was about 7% of follicular lymphoma, about 10% of all of diffuse large B-cell lymphoma, but he only found these mutations in the germinal center B-derived diffuse large B-cell lymphomas and not in other subtypes of lymphoma. He found that there was a recurrent somatic mutation in tyrosine 641, and that could be mutated, and that would result in amino acid changes to asparagine, serine, phenylalanine, or histidine. And they always found these mutations as heterozygous, and both the wild type and mutant EZH2 were expressed in these cells. What he found initially and reported in the paper was that the mutations were shown to be inactivated based on an enzyme assay that they did using H3K27 containing unmethylated peptide. And so when we talked to people and thought about this type of mutation, often when it's found in a kinase, would not be a loss of function mutation. And so we went on and investigated it further. And so we repeated the initial observation from Mark O'Mara's paper and can show very similarly that the wild type enzyme had very nice activity when you use the histone H3 peptide containing K27, and none of the mutants had activity. So instead of using this peptide, what we decided to use was the nucleosome, and a native nucleosome that we had set up for our enzymatic assays. And so when we did that experiment, you can very easily see that they were not inactivating, but they were active when we used native nucleosome as a substrate. So on further investigation, what we found was the following, that if you look at the non-methylated peptide, K27 peptide, and look for methylation activity with the wild type or the different mutants, you can again see in the unmethyl peptide, you see very high activity with the wild type and low activity with all of the mutants. If, however, you now take as a substrate a monomethyl peptide, you can see that the wild type has less activity and all of the mutants start to have activity. If you then look at the dimethyl H3K27 peptide as a substrate, you can see that the wild type is fairly poor at catalyzing the di to trimethylation, but all of the mutants are very active. So if you think about this mechanistically, we have the wild type enzyme that's able to catalyze very well the unmethyl to monomethylation, whereas the mutant is able to catalyze the di to trimethylation. And as I said, in these lymphoma cells, they're always expressing both the wild type and mutant. And so what you would expect is if you combine the two, that you would have a very high production, more than the normal, of H3K27 methylation. And so we performed this experiment biochemically where we did a mixing experiment where we had the wild type or mutant, and we looked at relative ability to form H3K27 trimethyl. And you can see that the mixture of the wild type and mutant, you were able to have the highest ability to form H3K27 trimethylation. So we also then wanted to look in cells, because you would predict that cells that had the mutation would have higher H3K27 trimethylation. And so if you take a panel of cell lines that either are wild type for EZH2 or mutant, and these are all lymphoma cell lines, you can see that they all express about the same level of EZH2, but the cell lines that have the mutation, you can see much higher levels of H3K27 trimethylation, and then, interestingly, very low levels of the dimethylation. So anytime you would predict that anytime that you form the dimethyl, their mutant enzyme is there to convert it to the trimethyl. And so these cell lines are all characteristically have very high levels of trimethylation and low levels of dimethylation. So then we thought this was a great program, an approach to take in, are these cells that have the mutation now dependent on the activity of this EZH2 mutant? And so we now have been able to identify small molecule inhibitors that are inhibiting, and these are the ligand affinity maps showing you the inhibitor selectivity of a typical EZH2 inhibitor that we've made. And again, the size of the circle indicates the potency of inhibition. So the larger the circle, the more potent the inhibition. And we've been able to make nanomolar inhibitors that recognize all of the mutations. So they're good inhibitors against all the mutation as well as the wild type. And they're about 100-fold less potent in inhibiting EZH1, but basically show no inhibition across other enzymes that are either the lysine methyltransferases or the arginine methyltransferases. Again, so a very selective inhibitor of EZH2. So then we looked as well in cells to see whether we saw selective inhibition of H3K27 trimethylation, and you see nice dose-dependent inhibition of H3K27 trimethylation, and basically no other methylations other than H3K27 are affected in these cells, again indicating that we're seeing very selective inhibition of EZH2 in cells. And so the next question becomes, are these cells dependent on EZH2? And so we've looked at both wild-type lymphoma cells and mutant-containing lymphoma cells. And you can see that those lymphoma cells bearing the EZH2 mutation are extremely sensitive, again, to inhibition of EZH2. And that was not because there was a differential in the ability to inhibit H3K27 methylation, because in both the wild type and mutant cells, we see nice inhibition of H3K27 methylation, but only in the mutant cells do we see inhibition of proliferation and death. So in summary, what I've shown you today is that the histone methyltransferases constitute a large and attractive target class for drug discovery. I've given you examples of two programs where we've been able to make inhibitors that are selective and show very specific effects in the MLL rearranged leukemia cells in the case of DOT1L, and in lymphoma cells that have EZH2 mutations for the EZH2 inhibitors. What I didn't have time to tell you about today is that these are not the only inhibitors that we have, but we've now been able to show very nicely that we can see, through cross-screening efforts that we have a nice biased library of inhibitors, and we've been able to make selective inhibitors now against multiple targets in this family. So I'd like to just acknowledge all of my fellow co-workers at Epizyme. It's really a terrific company. We've been in existence, as we said, for about 4 years now, and we have a great team. I'd like to just, as I say, give a shout out to a few people that have been involved in the programs. And that is Ed Alhava has done all the medicinal chemistry for the DOT1L inhibitor program. And the biology was done for the DOT1L program by Roy Pollack, Scott Daigle, and Carly Thurkelson. And for the EZH2 program, Kevin Kunst, again, was headed up the chemistry efforts on that, as well as the biology efforts, again, headed up by Roy Pollack and Heike Kilhack and Sarah Knudsen and Natalie Warholic. And I'd also like to acknowledge Scott Armstrong and a wonderful collaboration that we had for the DOT1L program. And so with that, thank you.